And common ancestry could be determined according to what derived characters a group versus previous groups has derived later on. So a derived character, it's a heritable character or heritable trait that a new group of organisms has acquired, but its ancestors don't have it. So, for example, the existence of mammary glands that define the mammals, or the presence of eyes, or anything that previous ancestors do not have, and that group has acquired through mutations and natural selection. That is called a derived group. So, for example, in plants, flowers versus cones. So that will be a derived trait. So that group that has acquired or somehow now has this derived trait, it's called a clade. And the word clade in Greek refers to branch. And a branch is monophyletic, or a monophyletic group, which literally means a single tribe group. And it contains its ancestors and all of the descendants. So from the word clade or branch, the word cladistics has evolved itself and refers to the inferences or all the hypotheses that evolutionary biologists have come up with to determine evolutionary phylogenetic relationships between groups. In order to determine these evolutionary trees that we'll soon find out, they're called cladograms, from the same root origin of branch in Greek, clade. Evolutionary biologists use parsimony analysis, which is really, in English, the simplicity rule. What is the easiest way to get from this ancestor or common ancestor to this derived trait, to this new group of organisms? So if you look at these three phylogenetic trees, or three hypotheses of how are these three groups of organisms that we just analyzed previously related to each other, which one is the simplest way from getting from a common ancestor here at the root of this tree to the three different types of organisms? So take a moment, turn the video off, and take a moment and count how many times have a new derived trait been acquired, right? So just count the red dots. All of these refer to derived traits that are existent in this monophyletic group or clade, but did not exist in their ancestors, right? So this branch here shows that they acquired wings, but their ancestors don't have it, and in this case, their sister group don't have wings as well. So which one of these three cladograms or evolutionary phylogenetic trees follows the simplicity rule or parsimony analysis? Is it A, is it B, is it C, why? How can you defend it? So now that you figured that out, you can tell that indeed, even though bats have wings, they are mammals when comparative to birds versus, in this case, dolphin. But you can change the group of dolphins with almost any other mammals that don't have wings, right? So lion or wolf or bears, etc. So as I mentioned, a cladogram, as its name says, it's a drawing of a branch or all the branches and then producing an evolutionary tree. And usually in evolutionary tree, it's rooted to an outgroup that is very far different than the branches that it represents. You'll see that in a moment. Wherever a line or a monophyletic group branches off, it's a node. And the two groups that branch out from that node are called sister groups. And they branch out because they acquired a derived trait. So that leads us to a quick revision of classification. So this is a very short version of classification, just to put things in perspective. So as we discussed already, even our own perception of life on Earth has evolved. And if we go back to the 18th century with Linnaeus, who is labeled the father of taxonomy, because he was brilliant by inventing the binomial system of taxonomy. He did have his flaws when it comes to evolution by thinking that all the species are fixed and they do not evolve. But his organizing skill were visionary, and we owe him this system of classification or nomenclature, binomial nomenclature. But in his time, we're very clear that there are two major kingdoms that are different enough from each other, the Plantae and the Animalia. Later on, with discovery of microscopes, which were actually before Linnaeus, but at the time, many fungi were considered very similar to plants. So, but then later on, we figured out that they're quite different than plants, so now they have their own kingdom. So this is early 20th century with Whitaker. There were 5 clear kingdoms that included, so it left animals and plants, and then it brought in new kingdoms such as fungi, protista, and monera. And monera refers to bacteria, because usually because bacteria are unicellular. So I want you to turn the video off and take a couple of minutes and try to come up with examples of monera, so bacteria, versus protista. What exactly are protista? And then maybe more than a couple of minutes, try to, of course, come up with examples of fungi and plants and animals, and make a very quick list of derived characters or very clear characteristics that differentiates amongst these 5 kingdoms. So what exactly makes fungi different than plants, even though historically they were studied by botanists? And the word botany means plant in Greek, so by plant biologists. So now that you have done that, you'll find out that actually fungi are heterotrophs. They cannot make their own food like autotrophs, that includes plants. So that makes them more similar to animals. But they feed in a total different way. And do fungi have cell wall? And if they do have a cell wall, is that making them more similar to plants? The fact that they're many times immobile makes them more similar to plants, and so on. So compare these major 5 kingdoms and give examples. And when we get to protista, protista are jokingly referred to the dump site of systematics, because they include a lot of organisms that are probably, some of them are fungi-like, and some of them are plantae-like, or plants-like, excuse me, and some of them are animal-like. But they have major differences to all of these 3. So they might feed the same like it. They might have, for example, chloroplasts. Algae are part of protista, but they are not plants, even though they are photosynthetic and they have chloroplasts. So what exactly makes them their own kingdom? What derived characters they don't have comparative to the other groups? But other protista include amoeba and paramecium and euglena and volvox and so on. So take a few minutes and just peruse through the internet and your textbook and try to visualize these groups of organisms and within their 5 kingdoms. So sometime in the middle of the 20th century, this group was discovered, Archaeobacteria. So in scums around the hot springs and hot vents of Yellowstone, this group was discovered. So, when they were viewed under the microscope, they look very similar to bacteria, and that's why they are called archaeobacteria. The reason actually that they were called archaeobacteria was the fact that they are very similar to archaic bacteria, or bacteria that inhabited Earth at the beginning of life on Earth. And that meant extreme habitats. That's why they're also called extremophiles. So they are adapted to very hot water, close to boiling point, which is extremely unusual because we know that both proteins and DNA denatures at very high temperature. But the reason that you were able to do PCR, or polymerase chain reaction, in molecular biology previously, before this class, was because we now have the DNA polymerase of Thermus aquaticus, one of the members of the now labeled simply Archaea organisms. Question for you, I want you to review The Making of the Fittest, where Shan Carroll, the author, explains why archaea are not considered bacteria anymore, and even though they are prokaryotes when compared to the eukaryotes that have a true nucleus with a double nuclear membrane, what exactly in their genetic makeup makes them more similar to eukarya, and what exactly makes them more similar to bacteria?